Check. Check, check. Ready to drop a beat?
Please do. Please do.
Not ready for it. You're not. We're at the hour, so we'll go ahead and get started. Sorry, first we'll take a little bit of a tangent because there are a number of first-year TVMH students looking for the QR code for today's speaker. Unfortunately, that wasn't produced in time for that, so I see most of your faces and I have a mental recollection. I think everyone's here, so we're going to give you a pass on the first one, so don't worry about that this week. I think most of you are here, actually, so that's good. Well, welcome to today's Pioneers in Biomedical Research Seminar. It is truly a great pleasure to introduce our speaker, Dr. Julia Mack. Julia and I share several connection points over our scientific careers, with the first, I think, being during our postdoc, our mentors. While Julia was being trained by Dr. Luisa Irulia Arispe, always trouble with her name, at UCLA, I received training from Dr. Victoria Bouch at UNC. And Luisa and Vicky shared common interest in angiogenic remodeling and the fundamentals of vascular biology. So while I was in Vicky's lab, we always enjoyed and admired the work out of the Luisa's lab, which included Julia's landmark work on the Notch pathway and arterial mechanosensing. I hate the Notch pathway, so I admired her even more for tangling with it because it's such a bear. but it yielded excellent contributions to the field, including a Nature Communications paper in 2017. Julia and I met for the first time at a conference in Spain, I think.
Oh, yes.
Wasn't that? It wasn't in Spain?
Yeah, that was an amazing conference.
That was an amazing conference.
That was amazing.
And our paths have crossed several times since then. It's always an encouragement when you meet an esteemed colleague in your field, and it turns out they're actually a kind-hearted and thoughtful human as well. So I've been enjoyed my interactions with Julia in that we share a real sense of curiosity and wonder with science. So by way of background, by the way, we do kind of long intros here, so okay. Buckle up. By way of background, Julia earned a BA in chemistry at the College of Holy Cross in Massachusetts, and then I think for the long term has moved to the West Coast to pursue a PhD in inorganic chemistry at UCLA. She spent a bit of time in industry contributing to several patents, I found out, which I'll talk to you about that later because that's really cool, and returned to academic research by joining the lab of Dr. Aurelia Rispe as a postdoc. And as I mentioned, she contributed to has continued to rise in the ranks at UCLA ever since, now currently serving as assistant adjunct professor in the Division of Cardiology in the Department of Medicine at UCLA. Her CV is quite impressive, so just to share a few highlights. Julia has an extensive publication record with several articles in very high-impact journals such as Science, maybe you've heard of it, Mason, ATVB, Nature Communications, Cell Stem Cell, Cell Metabolism, and the Journal of Clinical Investigation, among many others. I could go on, really. Her work has spanned a wide range of topics in cardiovascular health, including atherosclerosis and obesity, but she has maintained a central interest in the intersection between blood flow and the vascular system, and the steadfast focus on such an important part of vascular biology has been recognized by numerous funding agencies, including American Heart Association, the NIH, the Chan Zuckerberg Initiative, and several other competitive internal awards. Julia has served the scientific community in a wide variety of ways, including extensive service as a peer reviewer on several funding agencies and top-notch journals. Julia's rigorous approach to science has established her as a clear leader in the field, and therefore is with my highest enthusiasm that I ask you to join me in welcoming Dr. Julia Mack as she shares her exciting insights on blood flow and endothelial cell biology.
Thank you. That was a very kind introduction. But I'm really honored to be here, and thank you for inviting me and hosting, and for the opportunity to present my work today. So what I want to talk about today is actually a story of resilience, and why I'm talking about resilience. When I started my lab right around 2018, this is when actually NIH had initiated this Trans NIH Resilience Working Group. And the idea of resilience actually struck a chord with me. One, we want money, right? And if NIH is interested in resilience, then okay, I should be also interested in resilience. No, but it was more than that. It was this idea that, with everything in life, right, there's always challenges, and how you respond to those stressors or those challenges is how you return to homeostasis. And that's what resilience is, right? And I feel like resilience has become this buzzword in the media, but back in 2018, it was not a buzzword, at least I don't remember. But there's also really cool work that came out of ATVB, a paper by Gao and Gallus, exploring the role of endothelial cell resilience. And so this idea, I had done my work, obviously, in Luis's lab studying endothelial cells and blood flow. And I was like, well, blood flow is in this way, and really, it's an important aspect of our cardiovascular health and resilience. So my background being in chemistry, when I had come to Luis's laboratory and I changed fields, I went from material science to vascular biology. There's a long story there, but in any case, I fell in love with the endothelial cell, and I fell in love with biology. And what, coming from a different background, I noticed, very traditionally, I think in biology, we tend to look at and focus on risk factors and pathological mechanisms, right? What goes wrong? And then, how can we then fix it? But I also wondered, well, what about just studying what goes right? What about just studying what's healthy? And maybe we can identify then resilience factors, the things that make and keep cells or tissues happy and healthy. All right, so I'm focused in cardiovascular health. I'm in cardiology, although I'm not a MD, I'm not a cardiologist, but I study the blood vessels, and I study specifically the endothelial cell. And this is an important cell. Obviously, without the endothelial cell, there is no such thing as a blood vessel. So by definition, you have to have an endothelial lumen to be a vessel, whether it's a capillary, an artery, or a vein. All right, so it's well known in the field also that blood flow influences the endothelial cell phenotype. Wall shear stress is very important, right? That's the frictional force of blood flow that's going across the lumen of your endothelial cells in that vessel. And it can be different throughout the vascular tree. And it also will influence the endothelial cell behavior. All right, so I focus on arteries. I tend to focus on the aorta, the large vessel that's connected to your heart. And so for this talk, I'm going to focus on arteries, and that's why I'm going to talk about the range of shear stress in the human arteries. And what you can see is that there is quite a range of atheroprotective, meaning protective flow or good flow. This is actually, really large, actually. But in any case, this has to do with vessel diameter and all that. But atheroprotective or high arterial shear stress is something that results in a very healthy vessel, right? So you get low proliferation, low apoptosis, low production of vasoconstrictors, but you get high expression of vasodilators like nitric oxide, all very important. Versus if you have low shear stress or oscillatory flow that goes back and forth, that's why there's a negative there. If the flow is going back and forth, for example, or disturbed flow, you get the opposite. So essentially you will have low expression of the vasodilators, high expression of adhesion molecules, inflammatory mediators, and proliferation and apoptosis. So what signaling mechanisms promote endothelial resilience? Well, let's focus, why not, on this region where it tends to be protective. So in the aorta, for example, this is the mouse. You can see the range of shear stress by the color plot here. You have regions in the arch where it's very low, very high in the upper arch, where the carotids are. And then you have this region down the descending aorta that is kind of more pro-resilience. So this is more uniform laminar flow. So why don't we focus on this region of this atheroprotective flow? All right, so I always start with a video because I do a lot of imaging, as you'll see. This is a monolayer of human aortic endothelial cells that were cultured overnight statically, and then we're going to expose them to unidirectional laminar flow, 20 dynes per centimeter squared. So this is capitulating what happens in the human descending aorta. And I like to show this video because the onset of flow actually results in a whole bunch of cellular changes. You see a lot of proliferation. You see a lot of, I call pruning events, like that cell just popped off. You see a lot of proliferation. There's a lot of activity that happens. All the cells kind of, are ruffling and they keep their junctions, but they're changing their positions, they're changing their shapes, and they're responding to this onset of flow. What I'll do is now take this out further to 48 hours. And I like to show that, it's different, right? So there's not as much proliferation. The cells are more, they're not migrating as much. And they're a little more what we call quiet or quiescent. So that's where the term kind of quiescence comes. All right. So if I take the before and after for you, here's your static. And then on the right is after 20 dynes per centimeter squared for 48 hours. Something very simple. If you just look at the cell shape, if you look at what they look like in static culture versus what they look like after flow, there is a morphological adaptation. Endothelial cells elongate and stretch out in the direction of blood flow. And so when people typically, when I first joined the research lab, everyone's like, oh, you should have a cobblestone morphology. And yes, you should have a cobblestone morphology for endothelial cells if they're cultured statically. However, if they're cultured under flow, they're not cobblestone. They are elongated. They will stretch out and they align with flow. All right, so this brings me to just show some images. Again, like I said, I do a lot of imaging. This is, if you look at low flow, which is, I'm taking the lower arch of the aorta, you can see that it looks like that cobblestone morphology. So if you don't have high laminar flow, the cells are cobblestone shaped. In the descending aorta, where you have that high laminar shear stress, you have this elongated shape. So this is just calculating the aspect ratio. From lower arch to descending aorta, you get a significant increase in the cell body elongation. So aspect ratio of the cell. Now, in my lab, we like to do a lot of in vitro experiments. And so the beauty of this system, we use the Abidi pump system, and the Abidi system also comes with different slides. So chambers for us to seed our cells and expose our cells to flow. And I like this Y-shaped slide because it allows us on the same monolayer to actually have a variable exposure of shear stress. So we have where flow converges at the back end of the Y-shaped slide, we actually have a region of very low shear stress, about 4 dynes per centimeter squared. In that area is where these cells are not exposed to high laminar flow, and so they do not elongate with flow. So these look similar to the lower arch. So basically, we're just trying to recapitulate in our dish, right, in our slide, the morphological difference between the low flow or low versus high laminar shear. And in our high shear region at the back end of the slide, you get that beautiful elongated morphology. And so we can recapitulate the increase in the aspect ratio on this in this in vitro system. All right, so I was thinking when I first started my lab, and I was first thinking about ideas. You always take things from what you had done previously, right? So when I was studying the notch pathway, I was really mostly interested in flow, right? And what the story was in the work I did during my postdoc was actually asking whether notch was still important in the adult. It was really well known. It's important for angiogenesis and vascular development, but is notch still important in the adult aorta or arteries? And we found that it is. If you inhibit notch in the adult, you get the cells go back to being more proliferative. You actually get an increase in inflammation. So notch signaling is actually important. But that wasn't the part I was interested in. I was interested in this asymmetry I observed. So when we elongated our cells under laminar flow, right, there's a beautiful, the V coherence in white here to show the outline of the border of the cell. What I observed is that the notch full-length receptor would polarize or accumulate at the downstream end of the cell. And I thought this was so interesting. Why is it not symmetrically distributed across the whole cell? This is just an observation I had. In the end, we really didn't do much with that in the paper. We just reported this. I can also tell you something really cool is that if you reverse flow, notch goes to the other downstream end. So again, I don't know why. We kind of just left it there. It's all in the paper. I think some of it's in the supplement. But this stuck with me. So this was something that I observed that I was like, this asymmetry is cool. And so it made me start to think about if you have elongated cells, right, this high aspect ratio, maybe the two ends of the cell are different. That was all. And so I wrote my American Heart Association Career Development Award on that idea that maybe elongated cells would have a difference on the two ends. All right, so because my background was in chemistry and material science, what did I do during the pandemic when everything was shut down? I did AFM. And this again is in the supplement because no one really cares about it, but I think it's cool, so I show you. So basically I took our elongated cells and I did mapping across the surface. And I point this out because I like to show that endothelial cells are really nucleus heavy, meaning this is the height profile of the cell, this is the nucleus. And because when they stretch out, you get very thin on the edges. So I think this is something that we need to keep in mind. But what I did was also force mapping across the surface of the cell and looked at the two ends. And what I observed is that I saw there was a higher stiffness on the downstream end compared to the upstream end. So this doesn't really mean much, except for just to say that I started to observe by biophysical measurements that the two ends might be different. All right, we've done more experiments since then. We've used a dye called Lardin, and this actually incorporates into the cellular membrane and actually will give us, based off of its mission profile, an idea of rigidity of the membrane. And so what we see is that typically on these elongated cells, the downstream end of the cell tends to be more rigid. And so this is indicated by what we call the membrane polarization. Sorry, I'm thirsty. Yes, please.
Maybe you'll get to this, in which case.
Yeah.
But is there any suggestion that there's, in terms of this fluidity difference, that there's some change in the composition of the phospholipids that are being laid down in terms of?
Yeah. Well, and that's one of the things we're seeing with like measuring here. This is indicated of sphingomyelins. sphingolipids. We actually see there's accumulation of sphingolipids at the downstream end. So there is some asymmetry. We haven't looked at we haven't done a whole measurement of all the different phospholipids, but I will tell you we did lipidomics, right, shotgun lipidomics to look at the profile. And I can say that between static versus disturbed versus laminar, different flow states will influence a different profile of different phospholipids. I don't know where they're distributed, but flow will influence the composition of the cellular membrane. So besides we see a asymmetric distribution of certain lipids, we also see, what was interesting, like bundling or aggregation of F-actin at the downstream end. And this was something that, I put this on a poster a long, long time ago, and multiple people, including Ellie Zima, actually came up. She's like, please, I see this. What does this mean? So it's just something where we've all seen it, but we don't know what it means. And so we're trying to get at actually this, but there is an asymmetry to endothelial cells under flow. All right, something else that we see that's asymmetrically distributed is actually caveolin-1. And this is a really important scaffolding protein that form caveoli structures. If you guys, caveoli are the little caves. And it's important, caveolin-1 is required for formation of these little caves in the cellular membrane. And within these kind of invaginations, actually there's a lot of signaling molecules. So this also is kind of cool that we see an asymmetric distribution, more caveolin-1 at the downstream end. So I'm kind of pointing out these little, what I call tips. But I had to be careful and remove the word tip because people in the angiogenic field did not like tip. But in vitro, you can see it's really quite clear, right? These white regions at the downstream end that are very bright for caveolin-1. And so we're here plotting and calculating the intensity of that signal. And there's more caveolin-1 at the downstream end of the cell. Okay, so we've done this. We did a lot to work with people to help us quantify by segmenting and then masking to get essentially the fluorescence intensity of whatever we're probing at what we call these three regions, an upstream, the mid-body, and the downstream. But really what's all happening that I'm interested in is occurring here at the downstream end. This is also cavin-1, so I can also show you this is another protein that's associated with caveolin-1 is cavin. We also see that polarized. All right, so I think at this point, hopefully I've showed you that endothelial cells elongate with flow, and we're starting to see that there's an asymmetric distribution of certain things that are expressed in the cell. What I started to think about was these things kind of look to me like a domain, like almost like a button or something, right? As aggregation of F-actin and this caveolin-1 accumulating here. So we kind of, the way in the story we call it is caveolin clustering, because I don't know what it's doing there, but we're seeing a clustering of this protein at a specific location. Now, that's cool, but it's more cool if it's there for a reason, right? And so I asked, okay, is there signaling that might be happening there because of this aggregation accumulation of caveolin-1? And so very important function, as I mentioned in the slides previously, is nitric oxide production. Endothelial cells are great at producing NO. eNOS is the important enzyme that enables that production of NO. And it needs to be phosphorylated in order to be activated. And so we stained for caveolin-1, which we always see it, that's like downstream end. And then we stained for phospho-eNOS. And this is phosphorylated at serine 1177, which is considered the active form of the enzyme. And lo and behold, we see these like focal accumulation of this active enzyme. I mean, that's kind of cool. Like, why is something there? And there's a lot in the field because caveolin has been associated with actually inhibition of eNOS. But in any case, this is what we see. I mean, this is what we reported because this is what we visualized and we did it many times. So this got us thinking that maybe these are signaling domains. All right, so this is one of my favorite videos because it happened. We got these GCAMP transfected endothelial cells. So they're expressing this genetically encoded calcium indicator in green. And this is the first video we took of our monolayer. So I always show it because it's like, when you get that first video of something working. And I stared at this for a long time because right after getting this video, the world shut down for COVID. And so I stared at this video for months, if not years, and I still stare at it. So let me actually play it again for you. All right. So when I looked at it, I just thought it was so cool. So here you have, I hope you can see these sparkles, right? I call them sparkles throughout the monolayer. What I thought was cool is that the whole cell body is not lighting up. It seems to be domains or regions of cells that are lighting up. Not all the cells are exhibiting this behavior. It seems to be distributed across the monolayer. So I stared at this for a long time, and then I can show you a zoom in of one cell to really convince you that at that downstream end is where we see this signaling events. So we can calculate it by quantifying the mean intensity over time. And I think the video is pretty convincing, but I'll play it again for you. But if we look at, this region here, that's where all the activity is occurring. And it's really interesting that there is this oscillatory behavior. There's this influx of events that are occurring over this 15 minutes of imaging. So this elongated cell, caveolin's there. This is where enos, the active form of enos is. And this is where calcium is entering. Okay, so we have signaling happening there. All right, if we look across the whole monolayer, interesting, by segmenting, we only segment the full-length cells because we want to capture where the activity is occurring. About half the cells are active in the monolayer. And then if we look at those that are active, majority of the activity occurs restricted to this downstream end. All right, so this is a video to convince you that there is a mechanosensitive component to this. If you look at, remember that Y-shaped slide where you have very low shear where the cells are not elongated and cobblestone, right? You see very little activity there. And look where the cells are elongated and where you have high shear, that's where you see the sparkles. That's where you see the calcium events. So signaling activity is enhanced with increasing shear stress. And this is a side-by-side video view, and I think it's quite convincing here, right? On the right, the high flow, you see all the activity. It's playing, I promise you, on the left. It's just in the low flow, there's very little activity. So this is the same monolayer. These are all the same cells exposed to the same media conditions. They're just exposed to different shear stress profiles. And flow is influencing their behavior, not just their morphology, but also their signaling capability. And so here, that's that same video I just showed you. Here we're quantifying that we call the IOD, index of dispersion. This is a kind of a metric for us to show persistent activity. And so the persistent activity is all occurring in high flow at the downstream end of the cell. And you have much less activity in low flow. All right, so what we've also done is we've looked at kind of a correlation because we wanted to make a correlation. We believe that with higher aspect ratio, so cells that are more elongated, we actually see more propensity for having this downstream activity. And so in yellow is the calcium activity. So we always do our imaging in about 30 minutes. And so the yellow is essentially where the change in fluorescence intensity was occurring. So that would indicate where calcium was coming in. And so you can see majority of that activity is all restricted to these downstream end of the cells where the cell body is elongated. Okay, so this is a cool video that my postdoc did. We watched, because of course the reviewer asked, well, what about the process of cell alignment? So we did that for them. We monitored calcium activity during the process going from static to flow aligned. And so remember how one of the very first videos I showed you was that process of the cells elongating. And so we're going to do 48 hours here, but now we're looking at the calcium activity. And what I didn't get to say is that he was marking the little zoom in is the same cell over time. And so you can watch the process of cell body elongation. Look at that cell now. The same cell that he was a little zoom in is elongated. When it's elongated, it has the activity at the downstream end. And then it migrates, changes shape, and you don't have that activity. So there really does seem to be a correlation with the morphology, the shape of the cell, and the signaling activity. Even though flow is all there. And now look at this. This is 36 hours. You see how the monolayer now seems to be more kind of elongated, more uniformly. That's when you start to see collective signaling behavior. And I think that's really important too. And so now at 48 hours, you see that same cell we followed throughout the whole process is now elongated and you have activity. And so This is just showing you that calcium activity is polarized after the cell aligns with flow and elongates. And this is that cell where we've shown from 0 hours all the way out to 48 hours. When the cell body is not elongated, you get either kind of whole cell body calcium activity or none. But it's only when the cell body is elongated, this aspect ratio greater than 4, that you see this localized activity. So something about kind of probably cytoskeletal, right? Components that need to be set up for enabling all these signaling molecules to be at the right place at the right time. Yes?
So in the observation over time, the cells that are actually elongating and polarizing their response, their calcium transients, are actually moving in some way, right?
Yeah,
so
yeah.
Migrating,
yeah.
as well as responding in a static way to the shear. So do you know much about the differences in the adhesion of the cell surface that's actually against the substrate? Because the migration,
Yeah.
there's adhesion signal.
Yep. Integrins and all these things. Yeah. I haven't looked much, we haven't for the process, because I'm more interested in kind of homeostasis, right? After like the flow conditioning is what I've been kind of focused on. I think the process of migration, all that is really interesting. However, what I can say I've been looking at is actually distribution of other things like adhesion molecules, like integrins, after cell elongation. And we are seeing, I don't think I have the slide, it's work that is kind of more preliminary. There actually is an asymmetric distribution of integrins and focal adhesion molecules like paxillin.
I mean, eventually in the vessel, they have to stay still.
Yes, yeah.
So I'm wondering how that balance is achieved.
Yeah.
Boundaries of domains are very discrete.
I'm sorry?
The boundaries of the domains of flash look very discrete. So is the compartment vesicular, or I mean, how does it keep so?
The change in cell shape?
No, I'm
Yeah.
The boundary between the non-flash parts.
Yes. Yeah, no, that's really, we're looking at this now. So basically we're looking now at laminar versus disturbed flow. What I can tell you with disturbed flow, the whole cell body lights up when you have this flow that goes back and forth. The cell shape is cobblestone. And what I think we're trying to get, something is restricting in the laminar flow that's unique to restrict the calcium activity to one place and not spread to the cell. And we think, so this is very preliminary, but we think it has to do with the ER morphology. We're seeing ER tubules in the elongated cells that stretch out, whereas in disturbed flow, we see more ER shape or sheets. So we're thinking there is something that's restricting. It is very distinct. I don't know exactly what, but we are seeing something that seems to be a dividing line. We're trying to get and understand what is going on, but that's a great observation.
The flow going on in your model is continuous.
Yes.
So that's slightly different than what we have.
Like pulsatility, yes.
Yeah.
We've done pulsatility, we see the same, yes, we've done that. Because we got that question many times. Yeah, thank you. Okay, so let me move. All right, so I hope I've convinced you that these signaling domains are there. What we wanted to make a statement about was that they were mechanosensitive. And so we did a very simple experiment. Here's the one flow elongated endothelial cell. We're just looking at the presence of these, calcium oscillations or calcium transients. And then what we're going to do is we're going to stop flow. We just literally stop the pump. Calcium activity goes away. And then we're going to, for 30 minutes, we record no calcium activity. And then we're going to start flow again. And the activity comes back right where it was before. So this was an easy way, a very simple experiment, right? But an easy way to say that this signaling activity is mechanosensitive. It's requiring the presence of flow in order to continue. Okay, so then we did an experiment where we asked I was curious, we asked ourselves whether or not cell body elongation would be sufficient. I kind of knew the answer. I figured it wouldn't. I was pretty convinced that it's mechanosensitive. But in any case, we had access to these pattern slides. So we could actually get the endothelial cells to elongate, just like they do morphologically, but in the absence of flow. So basically they stretch out on these lines, on these patterns. So we get the same aspect ratio as flow aligned. And this is interesting though. If we looked at caveolin, we actually do see that caveolin 1 in the elongated cells without flow is still polarized. We tend to see, I think this has to do with just like, distribution of things when the cells elongate. And we see that there is caveolin either on the left or right side because there's no flow, but it's not one end. So I think flow gives it some directionality. Phosphoenos is not polarized, but that's not to be, is not unexpected because in a static state, we're not seeing this calcium activity. So elongated no flow versus low flow versus high flow, we all know that phosphoenos goes up. We know that there's more active enzyme and there's more nitric oxide production. But what hadn't been reported that maybe there's focal locations, subcellular locations where enos is preferentially activated. Okay, so then I wanted to ask, well, how is calcium coming in? I'm not an ion channel biologist, but we are pretty convinced it's coming from outside in because if we add EGTA to our media, we don't see the calcium activity. So we think there's got to be calcium coming from outside the cell in. How is it getting in? Well, I did a few Googles during the pandemic time when I couldn't go into lab. And I actually did a Google search for, I think, calcium sparkles. And this paper came up. They called them sparklets, TRPV4 sparklets. And it's a beautiful paper by Swapnil, who still works on TRPV4 out of Mark Nelson's lab out of Vermont, where they reported that TRPV4 is important for vasodilation. And I was like, whoa, I think this is exactly what we see in vitro. That's kind of cool, right? When you see something reported in vivo first, and then you see that you actually observe it in vitro. So meaning your system is working. That's always nice. So, and I'm sorry, what I meant to say is that GSK205 is a specific TRPV4 antagonist. So we can block TRPV4 activity, even though TRPV4 channels are present. We can block their activity and that activity at the tip goes away. So calcium activity goes away. So it's coming in through TRPV4 channels. Now, we stain for TRPV4. Now, I thought, oh, that'd be super simple if TRPV4 is just accumulated at the downstream end. It's not. This actually, I think, gets the story more interesting. TRPV4 is not differentially expressed between low flow and high flow. It's not, it's not aggregating at the downstream end where we see all the activity. It's just ubiquitously expressed across the whole cell. And it's not mechanosensitive in the sense of things like enos goes up with flow. TRPV4 does not care. So it's always there, but why is it active with the laminar flow and why is it active at a certain location? And so we went back to caveolin 1. And actually Swapnil had also published a paper in lung that caveolin modulated TRPV4 open probability. And I think that's actually what's happening in our case as well. So again, and his work was in vivo. So again, we're showing in vitro recapitulation of what was also reported. So what we did was proximity ligation assay, and we just looked at TRPV4 and caveolin 1, and we find that it's physically associating at the downstream end. So there's more physical association under high flow, which makes sense to us. And under high flow, it's more at the downstream end. So we think that this domain where we see this aggregation of caveolin is enabling a higher open probability for, and probably other things, lipids, actually, the question asked earlier, I think is modulating TRPV4 activity.
Normally primarily ligand gated, and if so, what's its ligand? Or how is
there's multiple domains. There's a PIP2 binding domain. And there's a bunch of things that have been shown, like from Swapnil and Mark Nelson and Osama Haraz. The team has reported a bunch of things that modulate its activity.
Those are internal ligands.
Yes, those are internal. Yeah.
Internal. So they're not, if there isn't a secreted ligand.
Not that I'm aware of.
So how is it actually activated in complex? Is there a mechanical declination that's necessary with the anchoring?
Yeah, I think it's probably not just caveolin. I mean, it could also be that it's in caveoli structures. That's also a possibility that hasn't been shown, but there's been talk about that. It's a great question. And I always, whenever I see the ion channel biologist, I ask these questions because I don't know. But yeah, great questions. Okay. Yeah.
Over there. Okay.
Yeah, sorry.
Do you have the Z resolution to determine if those channels are facing the flow or if they're facing the substrate?
Yeah, where they are.
Yeah.
They are, they're expressed all over though. They're not just on the surface of the cell. Yeah.
So there's even in your first super res where you have the active, there's no polarity in the Z dimension or anything.
I mean, the endothelial, like, yeah, I know it gets so thin. It's very hard to actually observe the Z at those edges. Yeah. But I don't think TRPV4 is only expressed on the surface because I see it also like on the basal membrane. All right, so cholesterol, membrane, lipids. Oh, yes.
Maybe you answered this already. What liquid are you using for your flow? Are you using blood?
No, we're using cell culture media. Yeah. So this has 10% FBS and VEGF and all the rest. So we keep everything kind of consistent so that, you know, static cells or whatever we use, what we use for passaging the cells is the same as what we use for the flow experiments. All right, so we wanted to mess with the domains. And it's known that cholesterol is important for caveolus structures. So again, this is hand waving stuff here. But in any case, cholesterol is a really important component of the cellular membranes. And it's been shown to use M-beta cyclodextrin, which chelates the cholesterol from the cell. It will collapse caveolus structures. So that wasn't the point of what the experiment was. We just wanted to mess with the domains. And the way we did this is you see under laminar flow, you have the polarization of caveolin 1. This is elongated, aligned endothelial cells. It just, we added M-beta cyclodextrin for 10 minutes. And when we do that, essentially sucks up the cholesterol and it also collapses that, that polarization or, you know, eliminates that polarization of caveolin. And so that was what we wanted to do. We just wanted to mess with the domains. And so when we did that, we looked at control versus cholesterol depleted. You see a reduction in nitric oxide, and we don't have as much activity. We had actually no activity. However, if we activated TRPV4 with GSK101, intriguingly, the activity still came back, meaning that TRPV4 is still there. So this is all in the presence of flow. It's just that when we sucked out the cholesterol from the cell plasma membrane, it messed with the structure and function of the domains. So that's all we wanted to show. Okay, but now I need to get to what's the reason why we're interested in this. So we inhibited TRPV4 activity. So this is, you know, one thing that we keep on seeing is this calcium activity that's localized. So we took our flow elongated endothelial cells, and then in the presence of flow, we either added DMSO or GSK205, which would inhibit the calcium activity. So basically, we're keeping the laminar, you know, the atheroprotective flow, but we're blocking that calcium signaling activity. And when we did that, we did RNA sequencing, and we saw 2 different populations. And the thing that we started to observe was an inflammatory phenotype. So even though these cells are exposed to the atheroprotective and anti-inflammatory flow, if they don't have that signaling feature, they become inflammatory. And so what this, you know, in looking at our RNA sequencing data and all the genes, VCAM was one of the genes that were upregulated. This is a cell adhesion molecule that's involved in vascular inflammation. But the main kind of pathway was inflammatory response. And if we look at the upstream regulators, the dominant one was REL-A. So this is NF-kappa B. So basically, if we expose ourselves, these cells are under laminar flow, either in the presence of DMSO or GSK205. If you block TRPV4 activity, look at all this. This is nuclear NF-kappa B. So this is indicating that the NF-kappa B pathway has been activated. So that's interesting. So inhibition of TRPV4 activity, even in the presence of flow, results in inflammatory phenotype. And we also showed this by ICAM expression and E-selectin. Both of these are adhesion molecules that are expressed by the endothelial cell. And typically, these are not expressed very highly in laminar flow. But if you block the activity of TRPV4, they turn on. Nitric oxide is also reduced. And we see an increase in reactive oxygen species. So there seems to be this balance, this important signaling pathway that activates eNOS and somehow protects against inflammatory mediators. All right, so of course, in order to publish in a bigger journal, you need to do something in vivo. And I was like, shoot, everything I have is in vitro. What do I do? And so we did a very simple experiment because, wild type mice are easy to get. We basically just injected wild type mice with either GSK205 or DMSO, and we focused on the aorta. And what we saw is that there's an increase in VCAM1 and nuclear NF-kappa B. So this was just recapitulating what we did in our dish. All right, now, then to actually get at kind of a rescue, because I learned in biology, you always want to kind of end with a rescue. Can we boost the signaling activity to improve the response to inflammatory stimulus? So this is the idea. Can we actually, going back to where I started, enhance endothelial cell resilience by activating this signaling? So what we did is we did our flow-aligned cells. We took them off of flow, put them to static. Remember, this is where the calcium activity is not active. And then we hit them with TNF alpha, either with DMSO control or with GSK101. So either we're exposing them to an inflammatory agonist, but if we activate TRPV4 with GSK101, we actually saw an attenuation of the inflammatory response. So there seems to be some protection to these cells when they're hit with inflammatory agonists if we activate TRPV4 at these domains. And so here is DMSO, here's TNF alpha plus DMSO. You can see every cell has nuclear NF-kappa B. However, if we activate TRPV4, you have less. So we're attenuating this inflammation. All right, we also, because of course everybody asked me about piezo because piezo is cool. TRPV4 is apparently not as cool, but everyone asked me about piezo. So we did that experiment. And in fact, if anything, YOTA1, which activates piezo, actually augmented the inflammatory response. It did not attenuate it. And I kind of like this because I'm like, oh, I like TRPV4. Not everything's piezo. So this is GSK 101, where we're activating TRPV4, and you see there are certain cells that don't have the nuclear NF-kappa B, but with YOTA, we do not see an attenuation. And this is DCF, which is our reactive oxygen species. You can actually see there's significant reduction of reactive oxygen species when we're activating TRPV4 that you do not see. There's no difference between DMSO and YOTA1. So at least for vasodilatory properties, we think TRPV4 is really important. Yes?
If you do a conditional knockout of TRPV4 in endothelial cells?
We've done that now, yeah.
What happens?
So they get more atherosclerosis. We did a hypercholesterolemic model. They get more atherosclerosis, and but they're fine, meaning that we don't see major inflammatory response. They don't die. We have a whole survival curve. We've actually aged them out to 2 years. And we did echocardiograms last week. Cool thing we're seeing is that there's an increase in arterial stiffening. So there is some phenotype, but it's not really dramatic.
And that suggests that there is some consequence.
Yes, yeah. So this is the attenuation. So this is activation. We did this in vivo because we kind of need to, we always try to do in vitro and in vivo. So we began wild type mice where we hit them with LPS. This is a low dosage. Reviewers hated this experiment because they said I was going to kill the mice. I'm like, no, this is very, very low dosage, very mild inflammatory exposure with LPS. And then we either added DMSO or GSK101. And cool thing, just like in vitro, we actually saw a reduction in inflammation. This is all in the aorta. So we're always focused on the aorta, just so you know. And you can see that by staining on phos, where a reduction in VCAM1 and less nuclear NF-kappa B. But this one is cool, right? This is RNA of, we do endothelial flush of the aorta, so we can get RNA from the aortic endothelium. And look at that, NOS3 is up. So it really seems to be anti-inflammatory by this vasodilatory signaling that's really important. And because of that, I think there's a reduction in inflammation. All right, so cool, I have 15 minutes and I wanted to leave you with some unpublished data because all of this was published last year, where we have identified what we're calling a mechanosensitive signaling domain in endothelial cells. And potentially, this is what I say because I've tried to get money, but really I just think it's cool that we see the signaling domain. I'd be happy with just that. But in any case, I think it's TRPV4, the signaling domain is really important. All right, so now for the unpublished data, and I'll hopefully leave us with enough time for questions from the audience. But introduced this whole idea about laminar flow and this elongation of the cell body. And then we have this mechanosensitive signaling domain at the downstream end. But I want to go back to what I showed early on, where not all the cells exhibit the signaling feature, right? I think that's cool. And I mean, heterogeneity is also a buzzword, right? So I guess resilience and heterogeneity, I'm just going along with the train. But I think spatial distribution of things and heterogeneity across a system or a tissue is very cool. And this is, really well known in terms of thinking about liver, zonation, and all these things, right? It's really important spatial. I think also very important in the endothelium. All right, so going back, this is published, but I just remind you that in our cell segmented monolayer, half the cells are active, right? Of those that are active, the majority of them exhibit this localized activity. This is also cool. So this is physiological flow. This is the average flow in the human descending aorta, 20 dynes per centimeter squared. And we get that about 45% active cells. Now, if we reduce flow to 5 dynes, you have less cells that are active. If you go towards pathological flow, interestingly, you have more, you increase. Now, the experiment here that I need to explain is that these were all flow conditioned for 48 hours under laminar flow of 20 dynes, meaning the good flow, right? And then we either pull back flow to have less, or we push flow to pathological. And so the response of the endothelium to pathological flow from good flow is essentially to increase the amount of these signaling domains. I think that's cool that maybe this is a mechanosensitive, resilient feature of the endothelium in response to, to any kind of pathological condition. All right, then I was interested about the location. Remember how that sparkles at one of the first videos? Yes?
Really basic question, but what is pathological?
Yeah, so the like that atheroprotective flow is like this large range, right? Pathological is very high shear. That's actually can be damaging, yeah, where you can get also disturbances too. Pathological flow can also be disturbed flow, meaning back and forth or oscillatory. But high flow can be pathological in the vasculature. All right, so going back to the sparkles where I saw them across the monolayer, we've been working with a group out of Munich to actually look at whether it's random or not. Because that's always the question. Is this just random and the cells just, sparkle, whatever? Or is this controlled and organized? I'd love to think that it's organized because in biology, right? I think things are organized, but we'll see. But our preliminary data is indicating by marking the cells that have that persistent activity, we're looking at this Ripley's K function, we're finding that they are dispersed. And that's kind of cool. They're not random. They're not clustered. They seem to be dispersed across the monolayer. So I want to think that maybe these are like, sentinel cells or signaling cells that are like, marked across the monolayer. Yes?
You know that your reporter is uniformly distributed.
In terms of
It's going to report where it is, right? You're looking at fluorescence, and the reporter has to be in a particular place in order for it to report. Do you know that it's present?
Yes, it's present throughout the cell. Yes. So this led us to this idea of endothelial cell heterogeneity. So we did a simple experiment of single cell sequencing because that's available these days and everyone's doing it. So we did it. So basically we took our monolayer of human aortic endothelial cells that were cultured under 20 dynes. And Vicky did this in HUVEX under laminar flow. Yeah, Vicky did this. And actually I showed this, I was at UNC not too long ago, and I shared this data. We had some good discussions. But everything I think about is about these signaling cells. So I wanted to see, could we find them transcriptionally? And I think this data is kind of cool. So what we did here is, by UMAP, we looked at the different clusters and we found 3 clusters. Well, this is at resolution 0.1. And what I think is cool is if we increase the resolution, you start to see like this cluster 0 continues to subdivide, right? It continues to divide into multiple populations. But look at this one that is the asterisk under cluster 1. It remains even up to resolution 0.8, indicating to me that I think that these are transcriptionally unique, right? And I can tell you who these cells are, but this is for a grant that I'm writing, so that's why it's in a figure format. But what we're trying to say is that in the arterial endothelium, even in mice, it's been shown to have 3 clusters. And I think Vicky had like 3 or 4 as well. So there's something about 3 or 4 anyway. But this cluster 1 is something I'm really intrigued by. Are these our signaling cells? That's what I'm currently asking. And the things that are increased in this cluster 1 pathway are things that seem to be like, focal adhesion, cytoskeletal, ABC transporters, relaxin ECM receptor. I feel like these cells, what I want to call them, are more reactive or more responsive cells. This is all very hand-waving at the moment, but I will tell you, cluster 2 are proliferating cells. So this is very simple. These cells are all expressing Ki67. And actually, if you continue to subdivide them, you actually can see them by phase of the mitotic phase. It's actually kind of cool. But yeah, these are all cells that are proliferating. And they have the most genes that are differentially regulated. This cluster 0, the one that will continue to subdivide as we go to higher resolutions, actually, I want to call them kind of homeostatic endothelial cells. They express things that are associated with quiescence, including Notch, Wnt, MGP, suppression of BMP. There's 174 genes that are differentially regulated, but then, it can continue to subdivide. But this is the cluster that I've been focused on. And we're trying to figure out where to go with this work. And I'm going to end in like a second because I don't know. So I would love ideas. But I think these are our signaling cells. There's only 40 genes that are upregulated, total of 95. They're, endothelial cells are all kind of similar. They're not super transcriptionally unique and different from each other. These are all arterial ECs. But we're seeing things that I find interesting with collagen 4, for example, which is a really important extracellular matrix molecule. And they seem to be secretors, or there's a higher expression of collagen 4, ABC, like I said, these transporters, nostrin. This is endothelin pathway, also another really important pathway. This is a receptor for it. So these are things we're thinking about now, and I leave you with this because I don't have answers, but I'm very intrigued. And so we have time for questions, but I do want to acknowledge funding sources. Very thankful to the American Heart Association, who's been very kind to me with a Career Development Award. And this is the postdoc that is amazing in my lab and will be on the job market soon, so look out for him, Soongu Kong. And he is a powerhouse, and we have so much fun together because we both love watching ourselves. He has the same passion as I do for visualization. And he just was awarded a postdoctoral fellowship. So, and he's writing his P99 right now, which is a whole process. I never did that. And then I just want to thank some other funding sources and our collaborators, and thank you guys for your attention.
Thank you, Julie, for a very engaging and thought-provoking talk. So this is going to be motivated by laminar flow over sand.
Okay.
Which can cause sand ripples. So that's where I'm kind of like basing the analogy on. When you look at the distribution of these things, and you said they're relatively flat, but there is a nucleus.
Oh yeah, the nucleus.
Yeah. So when you look at the nuclear localization of those that are and are not flashing spontaneously, are those polarized towards one end or the other?
The nucleus is polarized towards the downstream end.
Great.
Yeah.
Because I then would imagine that as you change the viscosity and or the flow rate of what's flowing over it, you're going to change actually the pattern of where these sand ripples occur or something like that.
I know there's some cool work by Frago, I think is his name, who actually modeled. So it's all mathematical modeling of the endothelial cell nucleus being the biggest thing, and then the edges, it's like a tent, right? The modeling, though, he puts the nucleus right in the center of the cell, and it's not. And I think that's the problem here, because he was talking about membrane tension. And I think membrane tension is asymmetric here. But I think it has to do with exactly where the nucleus is located. And a question I got earlier about why the domain, right, why the calcium activity stops there. I think it also has to do with the nucleus, and other organelles, So also intracellular asymmetry.
Thank you. So related, and you've touched on, well, the aorta is a happening place for sure. I mean, for lots of reasons. You've already talked about pulsatile versus static flow. You've talked about turbulence versus laminar flow. But what about things like transmural pressure and, passive distension? So they're undergoing, they're getting crushed, right? But also they're being stretched. Every systole, the surface they're sitting on is getting bigger. So number one, do you think those are important variables along with flow? And secondly, is there any way to model this in 2D culture?
Those are great questions. Lisa, right before I was leaving, actually, she got a stretcher. There are like systems where you can seed your cells onto like kind of membrane that's stretchable. And Luisa was actually looking at that stretch versus shear. I don't know where that project went, and I was long gone. I've been just focused on frictional, on the shear stress it's a reductionist approach. And actually I wrote a grant not too long ago that didn't do so well, but it was all about just, reductionist system as a means of modeling something, right, in vivo. And it's a problem, right? Because we don't have all the components. We don't have stretch here. We actually also have a very stiff surface. The cells are sitting on plastic. That doesn't exist in vivo at all, and so that's a problem that we have. But I feel like I have to come to terms with that, and I can only do so much. And I would love to do 3D systems too. That's another thing. Actually, that's where I started in Luisa's lab. The whole goal of my background in biomaterials was we were going to design materials for cells so that we could watch their response to flow in vivo, in vitro in a 3D matrix. And we developed tools that were porous, and we published a couple of papers in biomaterials. But in the end, I found that doing biology is hard enough. So I've simplified it. Yeah, but great question.
Right here.
Yeah.
So I'm a fellow spatial localization enthusiast myself. So I'm curious, because if you do kind of like a cell-based either sequencing or proteomics, you're going to really lose a lot of the preferential enrichment. So right in the, however you call that, leading edge. So if you've thought about either doing some kind of calcium-sensitive apex pull down or something that really allows you to get at the signaling zone there.
That's the grant that just went back in to do proteomics, where we're using actually a probe that targets that location. That's en route. And actually cholesterol is differentially regulated under all these different flow states. So actually a different project, we're looking at what cholesterol is binding to under the different flow states.
So one of the students asked this, but I want to amplify it. Blood is not simply a fluid. It has stuff in it.
I know.
It has rigidity,
blood cells,
and encounters, generates friction. And I would imagine in microcosm, it's actually a sufficient substantial force, or at least significant, in addition to just the flow and the directionality. And particularly under conditions of like hypertension and those sorts of things. So have you thought about how to consider that in terms of the cellular responses? The viscosity, the friction, how that signals.
So the one thing we've used, like the cone and plate, is a system that often we'll use because that can mimic pulsatility and also disturb flow. And people will add dextrin to the media to increase the viscosity. But I've found that dextrin actually will cause cells to be inflammatory. What? Yeah, I haven't done that, but there's got to be something other than dextrin, because people, every time I see dextrin and they're reporting something, I'm like, dextrin alone will just make the cells inflammatory. So there has to be something better. Yeah.
In vivo, what does it do to that region of the aorta that is?
So these mice that are aging, we're actually looking in the lung now. I'll let you know. We haven't looked yet. Yeah.
My last question. The composition of vessels changes as you descend into the legs and the more peripheral you get. So do these mechanisms maintain, could they be applied to peripheral artery disease or aging, these different things? Are they maintained?
Great question. So cool thing, because everyone asked me, well, show me the in vivo relevance of this calcium signaling. We now have a collaboration with Valentino Greco. We're doing intravital imaging of calcium. And the paper that's coming out is on the capillaries, where we're looking at the capillary, actually the spatial distribution. But preliminary data that's going into my grant is we go deeper into the plexus, where there's the arterioles and the venules. And I don't know if I put the slide in here, but It's so cool. We see in the arterioles where the cell nucleus, you can tell that the arteriole, the cell nucleus is really elongated. You see calcium is restricted to the downstream end. Whereas in a vein, the venule that's just next to it, and the cell is not elongated, it's the whole cell that lights up. Yeah.
Awesome. Oh, I'm like mesmerized. So let's thank Julia one more time.
Okay. I did good.